All right, let's dive into a kind of detective story. But instead of a crime scene, we've got the cell. And the clues? Well, they're hidden in just mountains and mountains of biological data. Our mission today is to see how scientists crack the case of life itself using some of the most powerful tools in biology.
So first, you have to understand the core problem, which is just the sheer scale of it all. Modern biology, with its sequencing technologies, is pumping out an absolute tsunami of data. Every single day, this constant flood of raw information just threatens to completely overwhelm the very researchers trying to study it.
Which leads to the big question, right? How on earth does anyone make sense of this? How do you navigate this just gigantic ocean of information to find that one clue you're looking for? How do you turn all that noise into actual knowledge?
Well, the answer, just like in any huge investigation, is all about creating order out of chaos. And for biologists, that means building these incredibly sophisticated systems to store, manage, and search everything. We're talking about biological databases.
This is the main tool of the trade, the biological database. The best way to think of it is like a massive digital library, but for the life sciences. It's not just a messy pile of files either. It is a highly organized, highly structured collection of information that's been gathered from thousands of experiments, scientific papers, and computer analyses from all over the world.
Or, to make it even simpler, you can just picture a super-powered digital filing cabinet. Everything has its proper place, which makes it possible to find exactly what you need when you need it. So let's crack this thing open and see how it's all laid out.
Okay, so it's all about hierarchy. The entire database, that's the whole filing cabinet. Inside that, you have tables. Think of those as the individual drawers, each one holding related data, like, say, a drawer just for protein structures. Then, inside each drawer, you find records. These are like the individual file folders, one for each specific item, like a single gene. And finally, inside each folder are the fields, the individual little bits of info, like the gene's name, its DNA sequence, or where it lives on a chromosome.
So now that we've got an idea of how the evidence is filed away, let's actually follow a case. Imagine a scientist just discovered a brand new, unstudied gene. This is our person of interest. Our detective story really kicks off the moment its sequence gets uploaded to a database.
So the very first stop for our gene is a primary database, something like GenBank. You can think of this as the raw evidence locker. It's the original data submitted directly from the lab, no filter. But from there, other experts come in. They review, they curate, and they add notes. That's called annotation. This creates a much more refined entry in what's called a secondary database. These are the detective's official case files, validated, cross-referenced, and a whole lot easier to work with.
Okay, our gene is officially in the system. Now the real investigation can get started. It's time to, pound the digital pavement and start connecting the clues to figure out what in the world this mystery gene actually does.
And a researcher starts by asking some really basic questions. First, who is this gene related to? In the grand tree of life. What's the actual physical shape of the protein it makes? What is its day job inside the cell? And, maybe most importantly, is it connected to any diseases we know about?
So the first clue is all about family history. By comparing our gene's sequence to countless others in these databases, a scientist can find its relatives, its homologous genes, in totally different species, from yeast to mice to us. And this isn't just for fun. It tells us about the gene's evolutionary past and hints at how important its job might be.
Next up, we need a molecular mugshot. See, in biology, a molecule's shape is everything. It determines its function. Databases like the Protein Data Bank, or PDB, are huge collections of the 3D structures of proteins. Finding a match for our gene's protein in there gives our detective a massive clue about what it might physically do in the cell. So we know its family, we've seen its face, but what's its job? This is where pathway databases come in. Things like KEGG, which is basically a collection of detailed blueprints of the cell's inner machinery. A search here can show us if our gene is a known player in a specific process, like how the cell produces energy or communicates with its neighbors.
And this brings us to probably the most critical part for our health. There are databases like OMEM that are specifically designed to catalog the links between genes and genetic disorders. This is where a researcher can find out if any little variations in our mystery gene have ever been connected to a human disease. This is where the detective work can literally save lives.
And as you can probably guess, this database landscape is just huge. We've really only scratched the surface. There are specialized databases for pretty much everything you can think of. DNA sequences, protein structures, metabolic pathways, even how different drugs interact with specific molecules in the body. It's a whole world.
Now, with all these different specialized places to look, you might be thinking it's impossible to keep all the evidence straight. But, every good detective needs a headquarters, a central hub where all the clues come together.
And in biology, we have one. A great example is the National Center for Biotechnology Information, or NCBI. It's this massive, integrated resource that hosts many of the databases we've already talked about: GeneBank, OMEM, PubMed for scientific articles, and the tools to search them. It really is the one-stop shop for genomic detectives all over the world.
So what does this all add up to? When you take a step back, you realize that the very nature of discovery has changed. This is a fundamental shift in how science gets done, moving a huge part of the work from the wet lab bench to the computer.
I mean, just think about the power of that for a second. A researcher sitting in a computer can run a single search that uncovers clues about a gene's family, its 3D shape, its job in the cell, and its potential link to disease. And they can do all of that before running a single new experiment in the lab. That's incredible.
And the real-world impact is just profound. This massive shared pool of knowledge is what's driving so much of modern medicine and biology. As one of our sources put it, it helps to facilitate the fight against diseases, assists in the development of medications, and helps in discovering the basic relationships in the history of life.
So, we're left with this amazing reality. For the first time in human history, we basically have the case files on life itself open on our computer screens. With all of the world's biological data right there at our fingertips, the only real question left is, what are we going to discover next?